Ah, the beautiful story of actin and cell crawling. This is going to be great. So the interesting thing about actin, what makes it unique and different from the other components of the cytoskeleton, is that actin can take many, many forms. Intermediate filaments braid up into rope that provide structural stability to a cell. Whoop-de-doo. Microtubules are a little bit more versatile. They can either be spindle microtubules in mitosis, pulling sister chromatids apart, or they can be protein cables for intracellular transport. Great, but that's really all they do. Actin does an amazing number of different things in the cell. It's part of the cell cortex, it's part of the contractile ring, it's involved in many different phases of cell crawling, it increases the viscosity of the cytoplasm itself. Actin is highly versatile. And the nice thing about the cell crawling story is that we see actin in 3 forms being used in each of the individual steps of cell crawling, and that's what we're going to talk about now. So the reason why you're looking at one vertical line on the screen is because we're actually going to show this process in 2 different perspectives. The first perspective here on the left is going to be the cell in profile. So what I'd like you to imagine is if you're looking at a car, at an automobile, imagine that you're standing on the sidewalk next to the car and you're looking at the passenger side door. That's the perspective here on the left. We're looking at the cell from the side. Now imagine that same car, and you're lucky enough to have your own drone, and you're flying that drone above the car, and you're now looking at the camera feed as you look down on the roof of the car from above. That's the perspective here on the right. And so we're going to have cell crawling drawn in both of those perspectives. So in order to crawl, we really need to do 3 different things from the purpose of the cell. We need to reach out to the world in front of us. We need to anchor down as we reach out, and then we got to heave the entire cell body up to that point of adhesion. So those are the 3 steps of cell crawling. Reach out, anchor down at that reached out point, and then move the cell forward. And actin is going to be involved in all 3 of those different processes. So in process 1, in the reaching out, what actin is going to do is form something called lamellipodia. Lamellipodia. I think I spelled that right, but who knows? Probably missing an L. Lamellipodia is a fairly random actin meshwork that assembles here at the front or leading edge of the cell. Now, I've already screwed up just a little bit because that's not entirely right. The way that I just drew that, if this is the side view, if we're looking at the passenger side door of the cell, so to speak, the lamellipodia is going to be here. Now, if we look at the front view of the cell, the top view, the lamellipodia is, it's at the front, but it's actually pushing the cell membrane outward. And this is what I accidentally drew on the left just a moment ago. So it's this meshwork of actin where every single line is an actin filament, and we can see that they're kind of interwoven a little bit randomly to push the cell out. So the cell is extruding outward just a little bit here with these lamellipodia, and what's doing that extruding out is this actin meshwork, this lamellipodia actin meshwork. So that's the reaching out step. And that'll continue. The actin will continue to be made, and it will continue to kind of press against the base of the membrane. And so the deformation becomes pretty extensive. Lamellipodia reach out quite far. And I'll try to capture that. So this distorts the cell quite a bit. And it's all because of this actin pushing, this actin meshwork pushing against the base of the membrane. So that's step 1. Step 2 is the anchoring. Now from the side view, this doesn't seem to change much. We're just going to push out even more. But here, the actin has a very different shape. The actin is bundled, and it's all linear. It's all pushing outward towards that next surface. To really appreciate what's happening here, we need to look at the top view. And what's happening is those actin bundles are actually pushing out finger-like projections of the cell membrane. See, we couldn't see that in profile. But these organized actin bundles of straight linear actin is almost like actin battering rams that are pounding against the lamellipodia and pushing out these finger-like projections of the cell. So that's step 2 of cell crawling. And we see actin in its second form, in this filopodia form. Now, to get at the adhesion part of this, we really need to zoom in on just this portion of the profile. So let's say here's some lamellipodia, and here's a filopodia. That's the name of this structure, if I haven't said it already. Filopodia are these finger-like projections. And so in order to make the lamellipodia, we had actin as a mesh, and in order to make the filopodia, we had this bundled actin, as I'm drawing here. So that's a single filopodia blown up there on the left. And the actin is crawling along some surface, right? So if it's in a lab, this is going to be a treated surface of a polystyrene plate or a glass slide. If it's in the cell, this is actually going to be the extracellular matrix, and this is made up of its own proteins, fibronectin most commonly for this purpose, but it's made up of its own proteins. So we have proteins here in the green, and we've got, of course, proteins represented by actin in purple. Now, proteins are stable. The proteins of the ECM is what gives the ECM its stability, and the proteins within the cell, especially of the cytoskeleton, gives the cell its stability. I'll remind you that the membrane itself has the viscosity of olive oil. There's no stability in the membrane. It's very fluid and very weak. So if we want to adhere, we need to somehow have a protein that can bind to the actin directly, be a transmembrane protein, right? Leave the membrane, have an intracellular domain and an extracellular domain, and then bind to the ECM proteins here. So we need a protein that can bind to both of the protein scaffolds of the intracellular and extracellular environments, respectively. That protein is integrin. Integrin is the protein bridge between the actin inside the cell and the fibronectin outside the cell, of the cytoskeleton inside the cell and the ECM outside the cell. Integrin is a transmembrane protein, and it's doing this bridging duty. This is what's allowing the cell to stick to the surface it reached out to and not have the membrane spontaneously retract back into the cell body. Okay, so that's that. So actin in its second form, filopodia, right? No longer a meshwork, that was for lamellipodia, now bundled battering rams of actin that can push out these finger-like projections, giving us filopodia. And I'll take one quick detour here as well, talking again about integrin. Integrin is obviously a transmembrane protein. Part of it is inside the cell interacting with actin, part of it is outside the cell interacting with fibronectin. So we can take a brief moment to appreciate how integrin is made, because we know integrin's whole life story now, whether we realize it or not. There's a gene that encodes integrin in the genome of the cell. That gene has a promoter, a Tata box. The pre-initiation complex assembles at that promoter, mediator receives the signals to fire, and we get transcription. That gene is transcribed. That messenger RNA is processed, its introns are removed, it gets a cap, it gets a tail, it circularizes, and it leaves the nucleus and enters the cytoplasm, where a ribosome is recruited to the start codon by eukaryotic initiation factors. Translation occurs, A sites, P sites, E sites, and the protein begins to be made, but the first thing that comes out of the ribosome is, of course, a signaling sequence, because this is a membrane protein. And so SRP binds to that signaling sequence and brings that ribosome mRNA complex over to an SRP receptor on the rough ER. The protein is then transferred to a protein translocation channel in the ER membrane, and it is threaded into the ER lumen until a stop transfer sequence is translated. And that kicks the protein out of the translocation channel where it becomes embedded in the membrane. The rest of the protein is made until a stop codon is reached, and then integrin is captured and collected into a prevesicle by cargo receptors. Adaptins recruited it there. Adaptins also recruit the appropriate RABs and V snares and clathrin, and we make a clathrin-coated vesicle that buds off of the rough ER. It goes to the Golgi, ironically enough, by walking on microtubules. Eventually it makes its way to the cell membrane through vesicle fusion, and we've delivered integrin to the cell membrane where it can do this job we're looking at now. So quick detour just to give you a sense of appreciation for how much you've learned in this course and how all of these topics we've discussed do come together into a single story of life, which is pretty incredible. All right, so enough of that. So we've got step 2 done for cell crawling, right? We've got our filopodia extended and we've adhered. Now the last step is, as I've mentioned before, heaving that entire cell body to the point of adhesion. And to do that, we need actin in its third form for this story. And that third form is actin as the cell cortex. Most cells have a cell cortex made up of actin, not spectrin. Spectrin was unique to red blood cells. And so actin is in the cell cortex. The cell cortex is made of actin, and the cell cortex is the cage or the underlying scaffolding over which the cell membrane is draped. The cell has its shape and has its structure because of its cortex. So here what we have is myosin 1 grabs on to the cell cortex and through ATP cleavage literally heaves, drags, pulls the cell body forward so that it will roll towards the point of adhesion. And as the cell rolls to its adhesive point, that cell will have moved forward by a little bit, and it will have taken its first crawling step. Now, to crawl again, what do we need to make? Lamellipodia. We need to protrude and distort and extend the cell membrane outward a bit using this lamellipodia meshwork to push the membrane out. And then we have to make filopodia by bundling new actin that we can then use to batter and push these thin finger-like projections out of the cell. That's all fine and good, but useless unless we can anchor to our crawling surface using something like an integrin. And once we're stuck there, we can heave yet again through the actions of myosin 1, and yet again drag that entire cell body forward, yet another cycle of cell crawling. And this is the way that cells move. Ram out lamellipodia. That's your leading edge. Push, 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 push out filopodia to give yourself a point of adhesion. Let those integrins knock down and latch on to the adhering surface. And once you're stuck there, use myosin and the actin of the cell cortex to roll and drag and pull the cell body forward to that point of adhesion. That's cell crawling.